السلام عليكم. إن شاء الله راح نناقش Biochemistry of neurotransmitters بمحاضرة هاي. So طبعا the main resource is the lecture itself. You can go back to Mark's Basic Medical Biochemistry, the 6th edition. في 10 pages that nicely describe the process. لكن I advise you to go to this website as well. في good information in terms of anatomy, physiology, and biochemistry that you can refer to as well. So first we need to start defining and talking about the characteristics of neurotransmitters. You will notice that there are exceptions, but these are the main characteristics and features of neurotransmitters. So basically, a neurotransmitter is considered as such if it is synthesized and stored in a presynaptic neuron. The enzymes that are needed to synthesize these neurotransmitters exist in the presynaptic neuron as well. The neurotransmitter بعدين بيصير له release into outside out of the nerve cell, the presynaptic neuron, to the synapse following depolarization of the nerve, and it is usually associated with influx of calcium ions. So the calcium ion concentration goes up really high. Once it gets released, it can bind the postsynaptic cell to a receptor على the surface of postsynaptic cell. أو ممكن it can bind to receptors على presynaptic terminal as well. As a result of this interaction ما بين ligand, the neurotransmitter, مع receptor, بيصير في عنا signal transduction taking place inside the cell. The remaining neurotransmitter is inactivated. It's removed in order to go back to the relaxing stage. So there are different types of neurotransmitters depending on their structure. فعنا small molecule neurotransmitters وبرضه في عنا sub classification لهدول النيوروترانسميترز. فعنا biogenic amines زي الابنفرين, dopamine, histamine, serotonin, and so on. And these are synthesized from amino acids. في عنا as well amino acids that can act as neurotransmitters, as well as a modified amino acid اللي هو الجابا. Acetylcholine, and we'll talk about these three classes. بعدين في عنا ATP can also bind to a receptor inducing a signal. We're not going to talk about ATP. The second class is neuropeptides, and then we have gases like carbon monoxide and nitric oxide. We'll talk about nitric oxide only. كانوا in the past يعتقدوا بأنه every neuron has its own small molecule neurotransmitter. طيب كانوا يعتقدوا إنه في كل نيورون في له or group of neurons في لهم small molecule neurotransmitter only and a neuropeptide only. لكن it turns out recently إنه لا والله في عنا إحنا neurons that can contain more than one small molecule neurotransmitter. We have neurons that have a combination of small molecule neurotransmitters with neuropeptides or more than one neuropeptide as well. هلا كيف يصير في release ل one versus the other؟ هون research that is going on, and it has to do with the level and probably localization of calcium ion influx into the cell. So this shows the different structures of these neurotransmitters, the different classes. So we have these amino acids. We have monoamines like catecholamines, serotonin, histamine as well. These are the neuropeptides, so they are composed of amino acids, okay, and they are small peptides, and ATP as well. So there are different classes and they have these structures. So we'll talk about these molecules right now. Now, there's something important in terms of differences between neuropeptides and small molecule neurotransmitters, and you do need to know these differences. Okay, and we'll talk about some of them at least throughout the lecture. So in terms of neuropeptides, they are short chain peptides. They're composed of 3 to 60 amino acids. They can have large molecular weight. Small molecule neurotransmitters, well, yeah, you can tell from the name, they are small. The neuropeptides are slow acting versus these are fast acting. Neuropeptides have a slow response versus acute response for small molecule neurotransmitters. Neuropeptides have prolonged action, but small molecule neurotransmitters have short-term response. Neuropeptides can act on several receptors, so you can have one peptide binding to different receptors, but for small molecule neurotransmitters, they have their own specific receptors. Neuropeptides can change metabolism, but not necessarily for small molecule neurotransmitters. Neuropeptides can change gene expression. Here, most of them do not, but we've noticed that some of them do as well. Okay, but most do not. Neuropeptides are synthesized in the endoplasmic reticulum and the Golgi apparatus, but small molecule neurotransmitters, they're mainly synthesized in the presynaptic nerve terminal itself, mainly in the cytosol. There are some exceptions ورح نحكي عنهم إنه they are synthesized inside vesicles. Neuropeptides are synthesized in low concentrations, but for small molecule neurotransmitters, they are synthesized in high concentrations. Neuropeptides are found all over the neuron, but small molecule neurotransmitters are found at the axon terminals of presynaptic neurons. Neuropeptides are stored in large dense core vesicles, and I'll show you an image of these vesicles. Small molecule neurotransmitters are stored in secretory vesicles, and again I'll show you an image of those vesicles. Neuropeptides are released at low increase of cytosolic calcium ion concentrations, but small molecule neurotransmitters require higher cytosolic increase, higher increase in cytosolic calcium ion concentration. Neuropeptides have different sites of action than their origin, but small molecule neurotransmitters usually they act in the vicinity, and in the neighborhood of where they are released. Neuropeptides are not taken up by the presynaptic neuron, and they are not reused, but small molecule neurotransmitters can be recycled. They can be taken up again by the secreting presynaptic neuron, and they can be repackaged inside vesicles and released. Neuropeptides are more potent than small molecule neurotransmitters, and they are inactivated by proteolytic degradation or diffusion outside or away from the secreting cell. Small molecule neurotransmitters are terminated by reuptake, uptake by neighboring cells like glial cells, by diffusion, or by enzymatic degradation.